Hi friends, today we'll discuss the topic thyroid hormone biosynthesis and secretion. Thyroid hormone biosynthesis occurs in a series of regulated steps which involves um, iod- iodide uptake in the thyroid follicular cells, iodide oxidation, organification, iodothyronine synthesis, and storage and release of thyroid hormone. See, this is the thyroid follicular cells. This is the follicular cells of the thyroid gland, and this is the basolateral side, and this is the basolateral membrane, and this is the lumen inside the thyroid follicle cells, and this is the apical membrane. And the most important and the first step in the thyroid hormone synthesis is the iodide transport into these thyroid follicular cells. As we know, this thyroid gland contains 20 to 40 times more concentration of iodine as compared to this plasma iodine. So this iodide transport into the thyroid gland, it is an active process, and it involves the transport protein, that is a NIS, that is a sodium iodide synthetase, and as it is an active process, it depends on the sodium potassium ATPase. And as this NIS is an important transport protein, we should know some details about NIS. This NIS, it is a 643 amino acid glycoprotein, and it is encoded by SLC5A gene, and it also present in other sites besides the thyroid gland. These are the salivary gland, lactating mammary glands, choroid plexus, gastric mucosa, cytotrophoblast, and syncytotrophoblast. And besides this iodine or iodide transport, it also transport perchlorate, pertechnate, and thiocyanate ions. Another important factor, that is TSS, also that increases the transcription of the NIS gene and prolongs the half life of the NIS transport protein. So in this picture, this TSS, when it acts on the TSS receptor, by the cascade of the reaction, it forms the cyclic AMP, and this cyclic AMP activate the NIS. So in some condition, the NIS expression are decreased, like thyroid adenoma and thyroid carcinoma. So as the NIS expression decrease, there is decrease uptake of the iodide into the thyroid follicle. So there is, in the radioactive scan, there is a cold nodule because of decrease in uptake. Then coming to another transport protein in the apical membrane, one is pendrin, that encodes by the gene SLC26A4, and another is anoctamin A1, that is ANO1 or TMEM16A. So these are the two protein. One is pendrin, another is anoctatin, and this after iodide enters into the follicular cells, this iodide then it flows into the thyroid lumen by the apical membrane, by majority by the pendrin, and another protein is the anoctamin. Regarding this pendrin, it is present in the apical membrane, and this pendrin is also expressed in the inner ear and in the kidney. When there is mutation in this pendrin, that leads to Pender syndrome. It is an autosomal recessive condition. In this condition, there is a sensorineural hearing loss, and there is goiter may be present. And this pendrin gene defect is mostly or iodide organification defect. And regarding anoctamin gene, it is a calcium activated anion channel, and it also helps in the apical efflux of this iodide. That is then coming to iodide oxidation and organification. The oxidation requires two enzymes. One is TPO, that is thyroid peroxidase, and calcium dependent DUOX1 and DUOX2. This TPO is the heme containing major thyroid microsomal antigen, and DUOX1 and DUOX2 are the calcium dependent NADPH containing oxidase, and this DUOX2 mostly helps in the generate the hydrogen peroxide, this H2O2, which is the major factor for the oxidation of iodide. So these are the two enzymes. One is a TPO, that is thyroid peroxidase, and DUOX2. DUOX2 helps in the production of the hydrogen peroxide, and this another factor, that is the maturation factor DUOX2A. It helps in the maturation of DUOX2, which leads to the functions of production of H2O2. and with this help of H2O2 and with presence of TPO, that is the thyroid peroxidase, the iodide oxidized, and after oxidation of iodide, this iodine combines with the tyrosine residue of the thyroglobulin, and this forms monoiodotyrosine or diiodotyrosine. This process is known as organification. So the process of organification is the when the iodine molecule it combines with the tyrosine residue of the thyroglobulin, that leads to formation of diiodotyrosine or monoiodotyrosine. It is known as organification. And what is thyroglobulin? Thyroglobulin, it is a 660 kilodalton homomeric glycoprotein, and it is storage of inactive form of thyroid hormone and iodine, and it is stored as colloid. So in this colloid, this thyroglobulin is stored, and when it combines with iodine, it forms monoiodotyrosine and diiodotyrosine. Then after formation of monoiodotyrosine and diiodotyrosine, next step is the coupling reaction. Then coupling, when two diiodotyrosine are combined in the presence of TPO, that is the thyroid peroxidase, is formed the thyroxine, that is T4. And when monoiodotyrosine and diiodotyrosine are combined in the presence of TPO, it forms the T3 or the triiodothyronine. Then coming to storage and release of thyroid hormone, the weight of the thyroid gland approximately 20 gram, and 20 gram of thyroid gland approximately store 5000 microgram of the thyroid hormone, and this 5000 microgram thyroid hormone can sufficient to maintain the body system in euthyroid state for at least 50 days. And when there is a requirement of thyroid hormones in the circulation, so this thyroid hormone from this thyroid gland enters into the thyroid follicle cells by either by macropinocytosis or micropinocytosis. And micropinocytosis is the most common mechanism in human. So when there is a requirement of thyroxine hormone in the circulation, the thyroid hormone binding with thyroglobulin enter into this apical membrane of this thyroid follicles by the pinocytosis, and it combines with the inside the cells, it combines with the lysosome, and then by the lysosomal protein, that is proteolysis occurs so thyroglobulin are separated from this T4 and T3, and T3 and T4 hormone that exit into these follicular cells by in the basolateral membrane by the transporter MCT8, and this MIT and DIT, that is monoiodotyrosine and diiodotyrosine, then recycled by the enzyme dehalogenase 1, and so that the iodide is recycled into the thyroid lumen. To summarize the thyroid hormone biosynthesis, the initial step is the iodide and uptake by the thyroid follicular cells, and after iodide uptake into thyroid follicular cells, this iodide then enter into this thyroid lumen by the protein transport protein, that is the pendrin and anoctamin, and after entering into the thyroid lumen, undergo oxidation by the enzyme TPO and duox2, then the duox2 generates the hydrogen peroxide, and after oxidation the thyroglobulin combines with the iodine, it forms the monoiodotyrosine and diiodotyrosine. This process is known as organification. After organification, there is coupling, it forms the T3 and T4, and it is stored with combination of thyroglobulin. So when there is a requirement in the cells, this thyroglobulin combined T3 T4, it is released into it enter into cells by pinocytosis, and then the hydrolysis occurs inside the lysosome, and T3 and T4 then released in the circulation by the transport protein MCT8. So this is all about thyroid hormone biosynthesis.